Hey, what is going on? It is me, and it is February 1st, 2022. January is done with and gone forever, at least in this universe. So, January 2022 is no more. Alright? And February has begun, and it's 28 days long. So, we've got a lot to discuss in those days. You also might notice the current state of my video and the situation I'm in right now. I will decline to speak about how I got into this situation, though if someone is looking at this video years later and is trying to figure it out, then I'll say that good luck. It's not actually that complicated. I just have decided to record a video late, wanted to try something different with my lighting, and yeah, here we are. So this is me, and I'm about to talk about synapses. Yes, so yesterday I talked a good deal about the axon, the structures of the axon. I went into the Schwann cells that protect the axon from leaking sodium during the transport of the action potential down the axon, and the nodes of Ranvier went over those, how those are basically sodium ion refueling stations along the length of the axon, and a few other things regarding the neuron itself and kind of how the action potential propagates and how sodium channels progressively open up, how the refractory period of the neuron is affected by the sodium transmission and propagation into the axon, and how the motion of the action potential is unidirectional, meaning the action potential only goes down the length of the axon. It will not go backwards, and that's great because otherwise our brain cells would backfire. And, that doesn't sound really great. So let's talk about kind of the end of that journey. And it's not really the end of the journey, it's the end of the journey for the pre-synaptic neuron. Okay, so when a neuron is connected to another neuron or is connected to another electrical cell that's electrically stimulated, that's called the neuron that is sending the signal through the axon to the destination cell is called the pre-synaptic neuron. And less surprisingly, the cell that receives the electrical transmission through the action potential, and we'll talk about how that is mediated, but the cell that receives that stimulation and thus that information is called the postsynaptic neuron, which has dendrites. If it's a neuron that receives it, other cells can receive axonic signals through action potentials, but in this case we're talking about a neuron, so there's a dendrite of another neuron that receives it, and the dendrites are connected to other dendrites, are connected to axons, and so on. So anyway, so that's basically just a little bit of terminology to make sure we can talk about this in a doctorly fashion. So we have our presynaptic terminals, axon terminals. This is where the axon ends. Then this structure, the synapse, is also called the synaptic bouton, I believe. And boutons are, I think it's a medical terminology for like these bulbous type shapes that occur in all over medicine, in the medical field. You probably have a bunch of bulbous shaped objects within you right now. Don't ask me how they got there. But yeah, so your neurons have these bulbous shaped end parts called synapses and synaptic boutons. And within the synapse, you have these mitochondria, which are pretty much within any cell, right? They're the powerhouses of the cell, so they provide the energy to the cell to continue its cellular, the different cell cycles. It allows glycolysis to continue, provides ATP and so on, oxidative phosphorylation and all that good stuff. So, but those aren't the important parts. Those just allow for things to occur in the synapse, right? The different membranes, not pictured here, membrane proteins to be activated, which are done via ATP through the ribosomic activity. But the important part here or another important part, are these synaptic vesicles. So essentially, the synapse is not the presynaptic cell is not directly connected to the postsynaptic cell. The presynaptic cell actually has this junction, this space called the synaptic cleft, and depending on who the synaptic cleft is connected to, whether it's connected to another neuron or whether it's connected to a muscle cell, which is possible. Neurons are connected to muscle cells, and groups of neurons and neuron bundles via their dendrites are connected to muscle cells for the purposes of stimulating them, right? You need to tell your arm how to move. I need to tell my mouth how to move, and my breath happens automatically, so that explains that. But anyway, there's no direct electrical stimulation. What happens is these neurotransmitters, which there's many different kinds of neurotransmitters, but these neurotransmitters, this is the general form, are in these vesicles, and these vesicles fuse through excitation. They fuse through an exocytotic process. Remember, exocytotic means outside the cell or outside the cytoplasm. They fuse with the cell membrane of the bouton, right? And they release the neurotransmitters into the synaptic cleft. When the neurotransmitters are released into the synaptic cleft, depending on the type of neurotransmitter and the destination of the postsynaptic dendrite or postsynaptic cell, the neurotransmitter will typically bind to these channels on the postsynaptic cell membrane and through different mechanisms perform a certain action, right? Essentially, the binding will stimulate, enough binding will stimulate the action potential on the postsynaptic cell. And again, the same cycle will continue, where if a message is being sent, the message must be sent through enough neurotransmitter activation and buildup of the positive potential through the ion channels within the cell. So the ion channel opens up, sodium flows in to the postsynaptic cell. Again, the same process occurs as initially with action potential generation, where the action potential propagates along the cell membrane and so on and so forth. So that's how, one neuron affects another. So what is going on here at the actual ion channel? So I think a good way to look at this is in the specific case of the neuromuscular junction. Okay, so the neuromuscular junction is a synapse muscle fiber connection. Remember, synapses, not only neuron axon terminal synapses are not only connected to other dendrites of other neurons, they're also connected to muscle fibers. So in this case, the cellular communication mechanism is similar, but what happens is that the neurotransmitter that's contained within these synaptic vesicles is different. In the case of muscular stimulation, you have muscle cells are stimulated by acetylcholine. Acetylcholine is a very common molecule. Everyone probably knows about it, but it's probably one of the most well-utilized and well-known molecules or biological molecules in the body. Acetylcholine is connected. What happens is it is released from the postsynaptic cell through these channels here. It's connected, it's collected here at these ACH receptors, and then it activates the ACH receptor to open up and allow sodium in. Okay, so these are acetylcholine activated receptors. Once the sodium gets in, then the muscle cell is stimulated and it'll keep firing action potential, right? Because sodium will keep building up in the muscle cell, and then that will trigger a downstream flow of the charge through the action potential mechanism. We don't want this muscle cell to fire forever. And by the way, that's why it's called a neurotransmitter, because acetylcholine is transferring that action potential. But we don't want, we do not want to like, box ourselves in, right, to a specific situation. We want to talk about generalities, but also specifics. But we don't want to continue to fire endlessly. That's what I was trying to say, right? As long as the acetylcholine, right, which is this molecule here for reference, is bound to acetylcholine is bound to the acetylcholine receptor, what's going to happen is that more and more positive charges are going to flow in, and the firing is going to happen over and over again, and then you'll have some kind of disease. That is the basis of some kind of neurodegenerative disease, I'm sure. So these ACH receptors then have to be broken down at some point so that the gate closes. So what happens? How do you break down an ACH receptor? So you have something that floats by called acetylcholine esterase, right? And what acetylcholine esterase does, by the way, these receptors that we're talking about have a specific name that accept acetylcholine. They're called acetylcholine. They're called cholinergic receptors, as it's pronounced here. And cholinergic receptors, there's many different types in the body and many different clusters of cholinergic receptors in the brain, which we'll go over at some point later on when I go more into the brain. But for now, what happens is acetylcholine esterase enzyme comes by and it finds the acetylcholine that's locked onto this cholinergic receptor, which is in many cases this sodium gated channel, and then it will allow sodium to flow in. So we don't want that to continue forever. So the esterase binds to the acetylcholine and breaks it down into choline and acetate. So once it's broken down, the choline and acetate go back into the presynaptic cell and eventually, through more internal enzymatic activity, get recreated back into acetylcholine and get repackaged back into synaptic vesicles through several organelle actions, and thus finally they get turned back into acetylcholine. They get turned back into acetylcholine, which goes through the same process again. So there's lots of cyclical processes in this nervous system communication. Many channels not pictured here. A lot of these are relying on ion intermembrane or cross-membrane ion transport to work. And there's many receptors, there are many neurotransmitters, which we'll have to go into. And we'll have to also figure out why there's so many different types of neurotransmitters. Maybe one is based on the way muscles react, the amount of potential required there versus how another neuron reacts. I think that's a lot for today. I feel there's something missing, but I think I've covered a lot in late in the day in my allotted time. If you like this video, please like, subscribe, and do whatever else you guys do who like watching my science videos. Okay, and specifically on eternal life. I'll see you later. Have a good